Yeah. Sorry, actually, in middle last lecture was actually about I'm here like just 13 minutes. Yeah, actually, because of some reasons I disconnected. Yeah, we are discussing about recalcitrant. See, means I told like what is recalcitrant? Sorry, recalcitrant seeds. What are the examples? How we can conserve it? And we'll go next. Then after conservation, I will come conservation and evaluation further. What are the different types in the coming lectures? We have separate lecture for that. But another activity in the PGR is characterization and evaluation. There is always a question: what is the difference between characterization and evaluation? Characterization means it just characterize the plant based on the observable character, highly heritable character. For example, you take highly heritable morphological or physiological or agronomic character, take DNA markers. So highly heritable, you can easily score, then you can characterize that individual. Or plant height, or color, whatever it may be. The color, they are highly visible, and you can easily, based on that, you can able to classify the individuals. That is only characterization. Means you are using some easily scoreable and heritable trait for classifying the germplasms. That is only characterization. But evaluation means the description of the germplasm for a quantitative traits like multiple years, multiple seasons means it is for quantitative. Just remember, evaluation you have to do evaluation for quantitative traits, further classification of the germplasms, and it's an interdisciplinary. You should include the team, include all interdisciplinary scientists. Then you have to evaluate in a multi-location and a multi-year. Then you go, okay, this germplasm is having this trait, this plasm is having this trait. Detailed evaluation of the traits. But here, based on the heritable trait, we're just classifying. After that, you evaluate in a multidisciplinary conditions, multi-locations, multi-year, okay, and all the biochemical and resistance, whatever it may be, all the traits you can measure. That is called as evaluation. Then it is directly asked in the exam. Then what is the need and importance of characterization and evaluation? Why we need to go? First, to reveal the information for an assessment or maintain the original population. If you know the original character, then you can maintain. If you don't know any original character, then how do you maintain? That is the one of the important thing. Then to locate specific and unique trait, based on the characterization only, we can identify the desirable trait. Then we can estimate the diversity present in that collection. Either we can use for fingerprinting the genotypes, because nowadays varietal fingerprinting is taking boom to avoid any IPR issues or any issues that we will going to discuss in coming classes. To do that, to avoid that one, we are going for fingerprinting of the genotypes or taking the genetic purity. It also useful. Then recognize the duplicates, because if normally one of the very important problem in the germplasm conservation is redundancy. Means we are collecting the same species again and again. Means this duplication will in terms of economic it is very big problem. That's why we need to eliminate this problem. Means you have to avoid the duplicates. Means recognition of duplicates and identifying them and removing them. It's a very important task in the germplasm conservation, and it's one of the important activity. Through characterization, we can able to identify the duplicates and we can manage easily. Then we can determine the gaps in the collection. For example, having 100 set of diversity, like 100 set of plants, but none of them are, if you characterize 100 set, but none of them are heat tolerant or none of them are any tolerance, then you are identifying, okay, these are not, that these traits were not there in my set, and you will start again exploring for the heat tolerant lines. Like this means we are able to identify the gaps in the collection. Okay. That's why we need to go for characterization. Then another thing is facilitate the preliminary selection and germplasm for end users. If you know the characters only, then you can able to provide the end users for breeder. End users are breeders. Okay, I have identified germplasm for this tolerant disease resistance. Okay, you can use it. That's why it's a characterization and evaluation very, very important. And many times asked in the exam, what is core, what is mini core, what is core collection, what is mini core collection, that we will going to discuss in coming slides. Then there is a, I told characterization and evaluation. And what basis you will characterize and what basis you will going to evaluate. We should have something to classify. Means that is called as descriptors. That is called as descriptors. ಕ್ಲಾಸ್ ಇದೆ ಇವತ್ತು ಆಗಲ್ಲ ಬಿಡು ಕ್ಲಾಸ್ ಇದೆ ಹಾ ಕರಿ ಕರಿ then descriptor like we need some traits to evaluate and to characterize and classify the individual. That traits are called as descriptors. Many times it's repeated in the exam. It will definitely will going to repeat in SRF. Descriptor is an identifiable and measurable trait or a characteristic of a plant accession used to make a classification, storage, retrieval, and use more info. And there is a separate board like now where it is developed. Like another one is descriptor state. Descriptor and descriptor state both are important, and many times difference also asked. Descriptor state means the expression of the descriptor. For example, you take color is one descriptor. How much expression of this trait? Amber color, red color, white color, golden color. This variation is called as descriptor state. Please remember what is descriptor and where it is used. It is used in the characterization and evaluation. Please refer the descriptors of at least wheat, rice, maize, three crops. At least you prefer what are the descriptor characteristics of these crops. Very, very important. I will share on PDF. In there you can read all these things. Then, what is core collection? These are very, very important to know. Core collection means, for example, you have, I think in NBPGR, in rice, we have lakhs of collection. But if you want to use lakh, if you want to select some individual, how do you evaluate lakhs of collection? You want to identify some genotype for term trait. You can't evaluate all the 1 lakh sessions. Then what you have to do? Had to do, you have to develop from 1 lakh set to at least 1000 set, or based on your convenience, you make mini set. Means large scale collections having lack of reliable data trait and economic importance, which show high genotypic interaction, like sorry, uh, make some reasons to low use of genetic resources. As this point tells, you have a large gene set. You can't unable to screen this larger gene set. If you are unable to screen means you don't have any data sets. That's why the utilization of these germplasm is very limited. To, to increase the utilization of these genotypes, what we can do? We have to reduce the number of assistants that will going for screening. Right? How we can reduce? There is a one concept called as core collection. Okay, what is core collection means? Small set of individuals, small set of individuals represent entire genetic diversity present in the large set. Means you sample in such a way that if you select many population from this one, it represents the whole diversity of the large population. For example, you take 10% of whole collection. For example, you have 10,000 individuals. 10% means 1,000 individuals you are selecting. This 1,000 individuals will represent all your 10,000 individuals. Means 10% of whole collection will represent all the diversity present in the world collection. And who given this concept? Many times asked, Frankel has given this core collection concept. Then again, this, for example, this 1,000 also very big. Then you develop a mini core out of core. Okay. And all this can be development of core collection based on your characterization and evaluation. There are many methods to do this core collection. Either you go for diversity analysis, either you go for cluster analysis, either you go for PCA, either you go for some target based core collection, whatever it may be. First, you do diversity analysis by PCA or D squares, and you can use clustering. And please note it down. Many readers don't know, like many of the students don't know what is clustering itself. It's pathetic. Should know how we can cluster the genotypes, how we can identify the diversity between the genotypes. That is our duty. We should not ask others to help us. Okay. See, I have published one paper like recently in scientific reports. I will share that paper. Please read how we done clustering, how we evaluated the germplasms, okay, how we identify the heat tolerant genotypes, then how we did PC analysis to confirm this one, and how we did which traits are we have used. Everything in detail I have done in that paper. I will share that paper. Please read that one. Then clustering, there are clustering is means so grouping of the individuals. There are several bases of clustering. Hierarchical based clustering and non-hierarchical based clustering. In that again several came like came cluster grouping, D square statistics. There are several things are there. Please read all those things. It's very important. Many times it's asked in the exam. Okay? This is basic statistics because I can't cover everything in the class. It's up to you that should have broad knowledge about each and every topic that currently we are doing. That's a very important thing. Okay. Then, what are the advantages of the core collection? Core sets are actually provides a ready reference set to assessment for assessing redundancy. For example, you have a lot of species, but there most of them are duplicates. No use of maintaining them. That's why to avoid the redundancy, to eliminate the duplicate, developing core collection will help a lot. Then, which are highest priority for conservation? For example, some genotypes are highly threatened. You develop the core collection of highly threatened species, then you conserve it. That priority of the selection is based on your objective. This core collection help in the selecting the priority genotypes. Then, and you can able to decide adequate descriptors. Then, and we can, if you have a core set, how many descriptor you want to evaluate, you can evaluate. But if you have a large set, you want to evaluate for 10 descriptor, how many labors you want, how much time you want. That's why if you are developing a small set out of the main set, you can evaluate for how many number of trait you want. Okay? That's why it improves the utility of your germplasms. If you develop a core collection, it improves the utility of your germplasms. Then, and nowadays everyone is using, I think AM panel, they're all mini core collection only. Then after that, okay, you have done characterization and evaluation by using descriptor and descriptor states. Then you developed some core collection. Then you have to document it. How we can document it? See, in earlier days, we are giving labels, we are using herbariums, whatever it may be, but it's laborious. We can register, we can, okay, this one is, we can write logbook, we can maintain logbook, whatever it may be. Either you can tag them, whatever it may be, anything is documentation. That helps in the compilation, data analysis, helps in the classification, helps in the storage and dissemination of the information. That's why documentation of PGR is very, very important. But nowadays we are living in the AI era. We are living in the computer era. Why can't we use This computer application to document our genotypes or germplasms. Seriously, it is asked in the exam. Please don't neglect these points. Two mobile apps were developed by NPGR like GeneBank and PGR Map to uh, having more documentation purpose. They'll put all the information of genotypes in the web. If you want, what are the information you need, you just search it. Then you can select the genotype. You can order for assistant. For assessing the genotype, you can write a request for that. Means they are putting all the database in a internet so that everyone can access. And GeneBank and PGR Map, actually it is developed by NBPGR only. Please read about it. It's very important. It asked in the 2022 exam. Then there is GeneBank app is also there. Then PGR Map. It's normally what's around me. For example, you want to obtain a quick accession that have been collected and conserved in the gene bank for particular. You want from Rajasthan area, you want all the gem plasms. You can just go to that website. You just click on the Rajasthan area. Then what are the collection which collected from the Rajasthan area? It will be visible. You can based on it. You can choose. That were PGR Clim and GIS Gene Bank, PGR Map. These are the apps. It's available in Play Store. You can watch it. Then you can use it. Okay, it's up to you. Yeah. Then please nowadays we are using this computer based applications or mobile Android based application to document and to disseminate the information of the germplasm that they collected. Then wild species reservoir of the gene. Like then why we're doing all this? Because the germplasms are reservoir of genes of biotic and abiotic stresses, and many desirable traits have potential improvement. They are found in wild species, and utilization of these species is a very good for the planting. But when you look at the history, it's very disappointing. I told PGR, types of PGR, what is land races, absolute cultivars, all those things we have discussed. Activities of PGR we have discussed. Okay, we have conserved all the area. For example, you consider all the wild species of rice. You selected also. But if you want to utilize in a breeding program, the success story is very, very less. Why it is so? Why it is so? There are several problems in transferring of genes from PGR to desired lines. There are such problems are called as linkage drag and initial detrimental effects to allied material. For example, you want to develop a resistant Pusa basmati line. You're bringing a gene from some wild species. But if you transfer the gene because of linkage drag, some other quality traits also from wild species get inherited into Pusa basmati. What will happen? For example, it affecting the quality. Who will buy your Pusa basmati? That's why you should take care of all the things. You should avoid linkage drag to avoid the detrimental effect from the donor parent to your allied parent. And to do this, for example, if you done okay successfully done crossing and transferring and making back crosses to recover the recurrent parent genome to avoid minimum donor genome, it's very very taking. Means at least you need 10 to 15 years to develop a variety. That too, just a modification of a single gene from a recurrent parent. It's time taking, laborious, and that's why before bringing directly the donor into the breeding program, we should go for one technique called as pre-breeding. Okay, what is pre-breeding? This is many times asked in this. What is pre-breeding? What are the activities we are doing before breeding of a crop? That are activities are called as pre-breeding. What are these? For example, you collection either in situ or ex situ. Then you have to utilize in the crop breeding, either breeding and pre-breeding. Then you go for seed sector. So all overall activity we're all doing. PGRs, breeder seed technologies. That why are all together. But what are the activities? These activities are there now. They're all pre-breeding activities. We'll discuss in detail. Pre-breeding refers to all the activities designated to identify the desirable characters and the genes from a non-adapted exotic or semi-exotic material and effort to transfer these traits into intermediate. Please remember, I'm not telling directly into a light. Intermediate material. To develop advanced breeding lines, by crossing them into donors. Then use that advanced breeding lines into the breeding program. Okay? Then it is act as a link between PGR and the plant breeders. It's a bridge. The PGR scientists here identifying the important donors and breeders here. It's a bridge. So this bridge is called as your pre-breeding. Then why pre-breeding is required? Means current limited genetic base because of monocropping. Current genetic base of agriculture is very less and it's threat for our food security. And there is a reduction in the biodiversity and high genetic uniformity and effect of climate change because new diseases, new pests and extreme temperatures are rising. We don't have any resource to combat for it. Then there is a dynamic evolution of pests and pathogens. You have to develop continuously new varieties to combat all these stresses. Then that's why we need a pre-breeding. What are the activities? For example, you're having gene pool. Either in gene pool 1, gene pool 2, gene pool 3, we're having a good source. For example, you have a good source at gene pool 3. But if you cross with your elite varieties, then definitely all this progeny will be sterile. But how you will use it? That's why we need a pre-breeding. First, you evaluate all the wild relatives and identify the promising accession. Then you bring that to hybridization program using diverse wild species. Then develop intermediate lines. Then through back crossing, you develop intermediate lines. Then use those intermediate lines which are performing agronomically good with your desirable trait. Bring that to working collection. Then you develop a new variety. Means all these are identification, developing advanced intermediate lines. All those things comes under pre-breeding, maintenance of gene pool, collections, conservation, all documentation, evaluation comes under genetic resources, PGR. Then after that, your breeding programs are designated by involving all your advanced intercross lines with your allied lines. You make crosses, then you develop desired varieties. This is all overall summary of the pre-breeding. Then this is about pre-breeding. Normally, we use backcross methods are majorly used in the pre-breeding. MABB, marker-restricted backcross breeding, all advanced genomic-based breeding, speed breeding, all things we can use. We can integrate with this pre-breeding approach to faster development of intermediate lines. Okay? That I am not discussing again because we discussed backcross, we discussed marker-restricted backcross, we discussed all the advanced methods of improving the genotypes. Okay, all things we can use in pre-breeding also. Okay, then after you develop advanced lines, intermediate lines, then you use that advanced intermediate lines in the breeding program. Directly, instead of utilizing donor lines with the elite parent, you first develop intermediate line, then use that intermediate line in the breeding program. This is what the pre-breeding is. Activities of germplasm conservation and the pre-breeding. And ex situ and in situ conservation, I will share Usha Kumari book PDF. You read in detail. Okay. Then we will discuss in next class. We will discuss what is policies and guidelines related to PGR management. This chapter is very, very important. Thank you.